Okay, everybody, we're going to get started, and we're now on a really tight time frame because I know that when we get close to the end, everybody is keen to get back onto the road or public transport. So I'm going to start with our next session. Our next session, I'll start by saying that we have 3 speakers within the next hour. So based on that, I'm going to ask each of the speakers to try to maybe limit their presentation to 20 minutes so that we can sort of get through to this. But it's such a good quality of speaker, it's really hard to stop. But I will get a tingle on the glass so that they know that they've reached 20 minutes, and then I'll leave it to them to decide. So, Professor John Mamo, being bloody-minded about Alzheimer's, challenging the brain-centric view. And John is a John Curtin Distinguished Professor at the Curtin University and head of the Neurovascular and Metabolic Diseases domain at the Perrin Institute for Neurological and Translational Sciences. With more than 35 years of medical research, has led landmark preclinical and clinical studies in cardiometabolic and neurodegenerative disorders, including Alzheimer's. And I'm actually going to not cut into his time by giving out the full bio, but welcome, John.
Yes, thanks very much, and I'd like to indicate my appreciation to Chris and the executive team for inviting me here today. I don't know what's happening to my throat. Look, I lead a large team over on the West Coast. We're a group essentially of physiologists and clinical biochemists. We do a lot of discovery science, and that's translated to several clinical trials that we've got active in neurodegenerative area and also in some neurological conditions. But for today, I'd like to talk to you about perhaps a different lens in terms of what might be a trigger and amplifier for Alzheimer's disease. So just to get you into the feel of things, there's essentially the prevailing international focus of emaciology for neurodegenerative conditions is either through some sort of proteinopathy, which triggers the decline, through neuroinflammation or oxidative stress pathways, or macrovascular complications, as you would appreciate. And this dominates pretty much the majority of the research, and I might say also research funding. I want to talk to you about some upstream potential triggers for Alzheimer's disease specifically, and I'm talking about changes that might be occurring pre any other pathological conditions around the capillary integrity pathway. And what I'm particularly focused in our group's particularly focused in is changes in the blood-brain barrier integrity, changes in blood perfusion and the availability of oxygen to the brain tissue. And what will be the focus today will be around an interplay between the metabolism of amyloid associated with lipoproteins in blood and how that impacts on capillary integrity and risk for progression. So it's a different lens where we're trying to switch the considerations from neurons causing vascular damage to microvascular damage influencing neuronal function. So there's emerging evidence to support this way of thinking, but certainly not consensus. That includes contemporary genetic indications through transcriptomic data showing early endothelial changes preceding other pathological events. Some of the imaging modalities looking at permeability across the blood-brain barrier and how that relates to cognitive function, and also significant animal studies now showing that capillary changes are occurring well before you see any amyloid or tau pathology. My focus today is around peripheral metabolism of amyloid and how that interacts with the capillary vessels. So I throw this up first. This is 3 lipoprotein fractions in 2 subjects, the triglyceride-rich lipoproteins, chylomicrons from the intestine, VLDL from the liver, and then low-density lipoprotein, HDL lipoprotein. I throw it up to show you that there's buckets of amyloid associated with lipoproteins. And indeed, the studies say that probably 92 to 98% of amyloid in blood is lipoprotein bound as we currently measure it. And of that, about 67 or so percent sits with the triglyceride-rich lipoprotein group, including enrichment in the more toxic A beta 1 to 42 isoform. I'm I'm going to summarise about 15 years of work in this one slide, and we've shown that there are dietary, endocrine, we've been focusing mainly in diabetes, and genetic factors which influence the synthesis and secretion of amyloid from the lipogenic organs with the nascent lipoproteins. And this is direct, we think, direct translation of message with the protein being linked to the nascent lipoprotein, not processing of the precursor protein. So we've shown that there are drivers which promote the synthesis and secretion of amyloid associated with the lipoprotein moiety. And if you get increased flux through this pathway into blood, this compromises the blood-brain barrier. You'll see extravasation of the entire lipoprotein amyloid from blood into brain. You'll see neuroinflammation, neurodegeneration, and cognitive function. And I've captured some of our historic studies just to show you what I'm showing about. So here on the left-hand side is one single capillary. You can see the lumen in the middle, and I'm showing you the penetration or the extravasation of the entire lipoprotein amyloid moiety from blood into brain. In the middle frame is exactly the same capillary vessel, but you're seeing the co-location, the movement of the lipoprotein-associated amyloid with the lipoprotein moiety, and on the right-hand side is the merger of those two. And if you eventually see plaques forming, you can see very strong co-localisation of the amyloid plaque, top left-hand side, with these triglyceride-rich lipoproteins being indicated. The merged image is down the bottom. Probably our most compelling evidence came from a publication we did in 2021. We decided to genetically engineer mice to produce human amyloid only in the liver, nowhere else, just only in the liver. And we just let them go, and we wanted to see how they would proceed. And I'll just grab some of that. So firstly, the bottom three rows here show you amyloid burden in our liver-only amyloid transgenic mice at 6 months, 12 months and 18 months of age. 18 months is around 70 years human equivalent. And you can see clearly age-associated increased amyloid burden compared to our wild-type controls in the top three rows. We measure a whole bundle of things in terms of what's occurring in brain tissue. This is around neurodegeneration. The higher the bar, the greater the deterioration. And across the lifespan, you can see much higher neurodegeneration occurring in our liver-only amyloid transgenic mice in both the cortex and the HPF. We also measure cognitive function. We heard from Colin about learning being so critical. So this is a testing in the mice. Acquisition means learning, retention means memory. The higher the bar, the better. And we've just focused on the retention or the memory, if you like. Our wild-type mice are performing at about 125 of the latency measure in comparison to our liver-only amyloid transgenic mice with a very significant indication of cognitive impairment. And this is occurring prior to any frank or significant amyloidosis being realised. Now, I don't have time to explain to you why we decided to go investigate this drug called Probicol. It's been used in the cardiovascular space, particularly in Asia now, as a cholesterol-lowering agent. You might see the literature says it was withdrawn due to safety concerns. That's not the case. It was voluntarily withdrawn by Sanofi at the time because the statins were coming in, and they were much more powerful in terms of cholesterol lowering. It does have a potential to increase QT syndrome in about 2% of people who take this, and it, at the time, was also considered to be not so helpful in the cardiovascular risk assessment because it lowered HDL cholesterol, only to be demonstrated later that it accelerates reverse cholesterol transport, which is protective for cardiovascular evidence. We also have a new analogue of Probicol, which I won't discuss today. But our studies over the 15 years have essentially found the following. Probicol profoundly inhibits the synthesis and secretion of the amyloid with the lipoprotein moiety. It doesn't interfere with lipoprotein secretion, but it profoundly inhibits its secretion. And Probicol is a lipophilic drug. It sits in the core of those particles. And in plasma, it markedly accelerates the clearance of these particles containing amyloid, pushing them essentially entirely to the liver. That means you've got decreased vascular exposure, and we see with that a greater strengthening of the barrier properties. Probicol also talks to the endothelium and increases the tight junction proteins, which give it the barrier properties. And if you get Probicol penetrating into the CNS, which it does in small amounts, it's a very potent modulator of key enzymes involved in inflammation: superoxide dismutase, hemoxygenases, and glutathione peroxidase. And on top of all that, so it suppresses inflammation. On top of all that, Probicol is the most potent antioxidant compound ever discovered to date. So these are pleiotropic properties or polypharmacological properties which might be highly relevant to the whole evolution of Alzheimer's disease, whether it be by way of microvascular integrity or inflammation occurring centrally. If you're interested more in the nature of the drug, we put out a fairly significant review last year if you want to refer to that. So I just want to say some snippets in terms of what Probicol can do in some of our animal model data, and most of it is all published. So firstly, this is a dietary-induced model where you're promoting the synthesis and secretion of the amyloid with the lipoprotein moiety. This is a risk for leakage across the capillary vessels, 30-fold enhanced by SFA or cholesterol, completely normalised with the provision of Probicol. And if we go to our liver-only amyloid transgenic mice, if you focus to the right-hand frame, our wild-type controls, again, at around 125, our liver amyloid transgenics markedly down, give them Probicol, and they have preservation of cognitive function. Those studies essentially form the basis for us to secure Commonwealth funding through the Medical Research Future Fund. And about 3 years ago or so, we were able to launch the Probicol and Alzheimer's disease study. I know earlier when Catherine was talking about trials that are existing, I think she referred to this one as a lipid-lowering study. This is not about lipid lowering. This is about modulation of neurovascular integrity, and I want to emphasise that point. So I'll just go back. So the Probicol and Alzheimer's study, if I can get back. It doesn't matter. The Probicol and Alzheimer's study is a double-blind randomised control trial. Primary objective measures are obviously cognitive function or cognitive stabilisation using ADAS-Cog as the primary instrument. Secondary objective measures are amyloid burden, and also we do some MRI imaging to consider. It's indicated there. The power calculations are indicated there. Review of the power calculations suggests that will be significantly less than that was initially predicted. Thus far, we've recruited about a little over 100 patients to the trial. We've had 22 or so complete, and we've got now, I think, 4 people on open label extension, which was granted earlier this year. So the recruitment starts, we started off in Perth. We are now active in Hobart with Jane Alty as the lead PI there, and David Bunbaden also supporting that significantly. In Adelaide through Kathy Short's team, and very recently in Christchurch, New Zealand, through Nigel Gilchrist's Centre of Excellence there. So we're very grateful for their support in proceeding with this. So current use of Probicol is restricted to pretty much Asian countries for secondary cardiovascular risk reduction. So here's the countries where it's indicated. It's always in the context of hyperlipidemia. It's been around for a long time. And we thought it would be extremely valuable if we can access population health data set to see if this drug has any impact on dementia or Alzheimer's disease specifically. And early last year, we secured a population health data set, Japanese population health data set of approximately 23 million people. We spent the last 18 months going very carefully through that. And you have to appreciate that what we're exploring here is dementia incidence or risk in people who've got cardiovascular disease. So our comparator group are those who are on statins. So people with CVD, we took people who are either on statins or on Probicol or in combination with. And today will be the first forum where we will share that information with you. So firstly, a bit about them. So the people we start off from 30 years of age up, we're following them over a 10-year period, and we're reporting dementia incidence in people 50 years and above. And in that cohort, on statins only, the incidence of dementia was indicated at around 12%. In those taking Probicol only, half. And those taking propofol and statins, there was a bit of a potential additive effect. Meta-analyses suggest that statins can reduce dementia risk by about 0.2. There's some studies for and against, but that's what the meta-analyses are tending to indicate. And this reinforces that it's not necessarily around lipid lowering because statins are much more potent. We then have done an assessment against the odds ratio against the different statin use, and that's indicated here. So this is both the unadjusted and the adjusted, and hopefully you can see it from where you're sitting, but this is the odds ratio of propofol relative to statins combined or individual statins. And the top row shows any statin, and the odds ratio is 0.38. So it's a 62% reduction, OR, 62% reduction against statin use for dementia. And the individual statins are indicated, and the trends or the slight variation between them is not because of lipophilicity of the drugs that we can appreciate thus far. You can see that the odds ratio doesn't change dramatic when you look at Alzheimer's disease or AD and VD, suggesting that propofol may have beneficial effects in other forms of dementia. And this, I think, is consistent with our view, which is that the etiology of Alzheimer's is really reflective of a microvascular origin of disease, and that's why we think we're seeing similar degrees of efficacy. So I think that population health data begs the question of whether this drug might potentially be useful as a preventative. And of course, the group that we'd all want to look at are those with ApoE4 genotype indicated. And our first approach to this was, let's go back to our amyloid transgenic mice. So we grabbed them, we did another modification on them. We knocked out their murine form of ApoE, and we introduced either human E3 or either human E4. So it's a very elegant model for us. So again, looking just at the cognitive data, which is all I have time to share with you today. So our wild-type controls, genetically unmanipulated, are doing really well with their cognitive assessment, sitting at about 175. If you like our human controls, which is liver amyloid with E3, don't do as well. So our E isn't as good as mouse E in mice. As I showed you before, I didn't show you before, but if you have E4 as well as liver amyloid, the cognitive performance, again, is massively reduced. But here, if you give the HSHA, our amyloid transgenics, with only the E4 and propofol, again, we preserve cognitive function. So can you take this generally well-tolerated drug into people who have an ApoE4 gene and say this may protect you from dementia, not just Alzheimer's disease? Now, I want to talk a little bit about ApoE, because in blood, 98% of ApoE is lipoprotein bound, and its function in blood is to serve as the ligand to clear the very same lipoproteins that contain the majority of the amyloid. So it's the chylomicrons, the VLDL. Once the triglyceride is partially depleted, they pick up ApoE, and this serves as the binding ligand to receptors, principally in the liver, and it's the LDL receptor. ApoE has a very high affinity for the LDL receptor. And we know from our cardiovascular days that ApoE4 doesn't clear these lipoproteins as well as ApoE3, which to our mind says you might have amyloidemia associated with lipoprotein metabolism if you're E4. So we decided to test that concept in otherwise healthy subjects who were either E3 homozygote or E3, E4 heterozygote. And most of our participants in this study, again not published, are university students, so they're quite young. I don't have the time to show you that population. So let me explain what we did. So the patients, or these participants came in, we gave them on 3 different occasions, we gave them a different crossover meal challenge. They would either eat pasta with no fat, pasta with polyunsaturated fats, or pasta with saturated fats. And the amount of fat in the meal was pretty consistent to what we would eat on a Western meal challenge. And I'm showing you the fold change over the postprandial excursion up to 8 hours. So we take blood with them over time. And remember, diet will drive amyloid with the secretion of the lipoprotein moiety. So here we have the E3 And we're looking at that A beta 1 to 40 excursion. It goes up and it goes down, and it's complete in the postprandial period. So it's something that we sort of anticipated. Now look what happens when you look at the E4s. It's sustained up to 8 hours. And this is showing you the mean response within the treatment group. But when you look at the individual response and rank which meal did they respond most to, there was a synergistic effect of ApoE with the saturated fats, with a 2.3 odds ratio increase in postprandial amyloidemia. And then if you look at the 1 to 42 in the E3s, it goes up and it goes down. And when you look at the A beta 1 to 42 in the E4s, again, markedly elevated. Every time an E4 containing person has a meal containing fat through this pathway, you've got exaggerated amyloidemia. And I think that's consistent with our original hypothesis that this is challenging microvascular integrity. And I'm nearly there. I've spoken pretty quickly. So what are the core insights? Well, microvascular dysfunction, I believe, is increasingly recognised as a driver for risk and progression of neurodegenerative diseases, including Alzheimer's. We can't ignore that there's substantial peripheral metabolism linked to capillary integrity, and I think modulation of lipoprotein amyloid metabolism offers measurable risk reduction and may represent a novel preventative and therapeutic strategy. We have some problems. We measure A beta in blood, and I can tell you that neither high sensitivity Simoa or any of the ELISA methods or any of the coupled immunocapture MS methods, none of them measure lipoprotein amyloid. And particularly in 1 to 42, which sits in the core of the lipoprotein particle. We've been working on this for about 10 years. We haven't optimised it yet, but I can tell you there's buckets of amyloid that we're just not picking up. At the moment, we think at least a 4-fold greater abundance of A beta 1 to 40 in blood, and probably around a 30-fold increase in A beta 1 to 42, which is lipoprotein bound. Now, I'm not suggesting that any of our blood biomarkers are not sensitive or predictive tools for us identifying people at risk, but I'm saying we don't yet have the luxury position, as in cardiovascular disease, to understand how the metabolism of amyloid with different lipoprotein fractions might influence risks. So that's a significant issue for us going into the future. And as I indicated, I think it's really important that we look at ways of improving the potential efficacy of Probicol, and we've got a new drug analogue which we just hopefully will commence with comprehensive preclinical and safety and tox, which we addresses some of the limitations. Finally, I've got to acknowledge particularly the discovery scientists, Ryu and Virginia. Virginia's in the audience here. And I particularly want to point out Roger Clarnett, who was the first geriatrician to really come on board and support it. Very grateful to Kathy and her team, to James at UTAS, and most recently to Nigel and his team in Christchurch, New Zealand. Thank you very much.
In the interest of time, we might have just one question, if somebody's got a burning question. Dennis.
Some of the new agents, like the PSK9 agents, where they have other effects, are you seeing any effect on this finding that you had?
Yeah, well, PSK9 can have pleiotropic effects, not just around, cholesterol synthesis and how that might relate to synthesis of the amyloid. I think this is a completely different mechanism. I don't think it's really regulation of the genesis of amyloid from the precursor protein. So not necessarily apples with apples.
Thank you, Professor. It's been a fantastic presentation about the lipoprotein amyloid. It's really interesting. So now we continue now.